Okay, Krishna.
All right. Thank you, Veronica. Good afternoon, everybody. Welcome to the SCALAX, that is the Southern California local section of the American Chemical Society sponsored seminar by Professor Corey Hopkins. And before I introduce him, I would say a few words. First of all, we thank the local section activities committee, LSAC, for giving us a grant to promote seminars like this that would be of interest to everybody, including the senior chemists. And I happen to be, in addition to the local sections subcommittee chair on senior chemists, for senior chemists, I am also the subcommittee chair of the InReach committee of the National Senior Chemist Committee. And I just wanted to tell a very brief story. Actually, in March, I attended the San Diego Spring National Meeting, and we had two booths there, one organized by our chair who is sitting here, Veronica, and the other organized by the other chair, that is the national chair, the Senior Chemist Committee chair, Frank Romano. And then I was talking between the two, and for some time, there were quite a few people in the at the National Senior Chemist Committee booth, and subsequently we also joined me, Frank Romano, and then Lee Hoffman. And during our discussions with the visitors to the National Senior Chemistry Committee booth, I happened to see one young lady actually who was standing a little bit away from the booth, and then noticing all these senior chemists there, she had a smile, and then she moved closer to us, and then asked us what we are doing, and then we explained things, all that to her. And then I casually asked her what she is doing, and she said she is a graduate student at the University of Nebraska and working with Professor Corey Hopkins group. And she mentioned a very little bit about her work, including some that was very recently published on April 16th, I think, in Chem at Chem. And her name is Samaya Naren, and she actually while describing her work, she looked at me, especially because I walk with a walker, and she said, "My work will be very suitable for you because I know you are suffering from some pains and this and that and all that." And I asked her who your supervisor is, and then she mentioned Dr. Hopkins' name. And I was so happy because I know I was following his publications for a while. And I know he has more than 200 research articles, and which was cited by nearly 5,000 people. And I thought maybe I was also included in that. So that's how I really wanted to make sure that I invite Dr. Hopkins to give the seminar, because that's a topic on which most of us seniors are very interested in, definitely. So let me tell a little bit about Dr. Hopkins. He's a professor and interim chair in the Department of Pharmaceutical Sciences in the College of Pharmacy at the University of Nebraska Medical Center, and the director of the newly designated UNMC Center for Drug Design and Innovations, CDDI, that's what they call it. Corey completed his doctorate in 2002 at the University of Pittsburgh in the area of natural product total synthesis, and he developed a novel ring expansion methodology to make pharmacophore analogs of denacin. Dr. Hopkins moved to Aventis Pharmaceuticals in 2001 and later became senior research investigator in medicinal chemistry. In 2008, he served as he served as Associate Director of Medicinal Chemistry for the Vanderbilt Center for Neuroscience Drug Discovery, where he led the chemistry efforts on multiple projects that led to pharmaceutical partnerships and a first-in-class clinical candidates. He is the author of more than 110 peer-reviewed articles and an inventor on more than 25 patents. In addition, he has been named to Stanford Elsevier's top 2% scientist rankings for the past 8 years. Dr. Hopkins has expertise in medicinal chemistry and drug discovery. Dr. Hopkins Laboratory focuses on the development of chemical probes to elucidate the mechanism of therapeutic areas such as pain, Parkinson's disease, and cancer. And tonight, he is going to talk about the quest for painstakingly perfect medications, and we'll be talking, I think, quite a bit about the GIRK 1 by 2, which are the G protein coupled inwardly rectifying potassium channels. That's what the GIRKs are called. Now, without much ado, I would like to pass on the mic to Professor Hopkins to deliver his seminar. Professor Hopkins.
Thank you. Thanks for the introduction. And yeah, I was pleased when Samaya came back and said that she had talked to this and got the invitation. So I'm happy to be here. Make sure I am sharing the right one. Okay. Is that correct?
Yeah.
Okay. All right. Yeah, as was said, my lab's been working in a lot of different areas for a number of years. And I always tell people as a medicinal chemist, I'm pretty well disease agnostic. I don't have a disease area that that I particularly work in per se. I really look at interesting biological questions and where we can sort of make compounds to do that. So this slide just kind of highlights some of the variety of chemical matter and also receptor classes that we've been working on in the past about 20 years, from ion channels to GPCRs to kinases, really looking, collaborating with biologists to look at these interesting questions. And a lot of the work, at least as a medicinal chemist, you always want to see a project advanced, in this muscarinic 1 positive allosteric modulator, this VU319, was able to be advanced into phase 1 clinical trials for cognition in Alzheimer's. So it's a exclusively an academically driven project. So we discovered it at Vanderbilt, used NIH funding to get it to a point of sort of IND enabling, and then we were able to have a sort of an angel investor come through and give us the money that allowed us to go into phase 1. It's now since partnered, but it's very challenging to take these compounds into phase 1 clinical trials. It's just the cost involved, and even the expertise dealing with the regulatory can be very challenging. So it was a real pleasure to work on that. What I am going to talk about, I'm going to talk about 2 separate projects today, but they both deal with pain. And as said in the introduction, the first one's going to be on these novel GIRK 1-2 activators. And we've been interested in the GIRK ion channel for now probably the past 12 to 13 years with multiple different collaborators. So I do want to just start off and show this so I don't forget at the end. And this is Samaya is here, but other very talented collaborators that I've had in my lab, and Anish is now at Vanderbilt, Swagit's at Northwestern, and Lauren was an undergraduate in my lab who's now at Cal Berkeley. And then I'm a medicinal chemist only. My lab does not do any of the biology. We're not chemical biologists, so I need to have really nice collaborations in biology. So it probably doesn't need to be said that treatment of chronic pain remains an unmet medical need. Chronic pain affects about 40% of the people in the United States, significant healthcare costs. And you have the typical, the NSAIDs, the anti-epileptic drugs like gabapentin, you have opioids. They all are variable and somewhat unimpressive efficacy. And they also have significant side effects, particularly, abuse potential, tolerance, dependence in the case of opioids, which are still the most widely prescribed medication, although in many chronic pain instances, they do not work very well. But if you look at this stat at the bottom, which is pretty striking, so when pharmaceutical companies and others have looked for pain indications that have low abuse for potential, there's only about 5% that are in clinical development that were successful. But if you look at the high abuse potential, about 28% are successful. And the high abuse is the opioids. So the opioids and derivatives of those tend to have better success development. However, there is a significant NIH push towards getting away from those types of drugs. So some of the existing chemical entities that are in here, you can kind of see on this this table here. And really, it's pretty interesting to see this efficacy of several approved chemicals. And this is the number needed to treat, so NNT. And that means the number of patients needed to treat to achieve at least 50% reduction. So ideally, one patient. You treat one patient, you get 50% reduction. But something like capsaicin, which again is not a major pain medication, although you can get patches and such. But even something like gabapentinoids, you still need about 4 or 5 patients to do that. So there's still a lot of room to be had in development of pain medications. Pain is challenging, right? It's not it's not something that can easily be diagnosed, and everyone's pain is different. Think back in before the opioid crisis really came out, there was a big push to make pain a vital sign, the 5th vital sign. It's also the only subjective vital sign. You can't measure it. So the scale of 1 to 10. So it leaves room for misdiagnosis and also abuse. We're looking at these sort of peripheral nociception and brain modulations in some of these areas. So you have the supraspinal cords, nociceptive input, output. The dorsal horn synapse is very key in the spinal cord, and that's really the dorsal horn ganglion is some of the areas we're looking at. And then you can see a lot of these other sort of receptor classes that are being looked at as well. So again, the dorsal horn is an integrator of peripheral nociceptors, and these are due through a lot of these. These are trip channels, P2X and calcium. You have NAV and KV ligands, and we're kind of in the KV, and then you have GPCR. So a lot of variety in the receptor classes, but still not a lot of success. And we're looking, to go through the NIH, and they have a lot of, direct funding. So they have this HEAL initiative, and they looked at a lot of different targets, whether it be targets with FDA approval for pain, targets with prior approval but not for pain, so the cannabinoid receptors, and you can see down here sigma-1 receptor, which I'll talk about in the next project, and then new drugs, no approval history, and novel to pain. What was exciting recently was Vertex announced the positive results from the phase 3 for this VX548, which is a NAV 1.8 compound. And then even more recently, earlier this year, it actually was able to get approved. So this Jernavix or sustrogene is approved. It's interesting to talk to some of the clinicians in Nebraska, and they're even seeing some individuals that are refractory to this as well. So there still is this major medical need. So what we're looking at is we're looking at these inward rectifying potassium channels. There's a large number of them, so a total of 15. Not only are they 15 distinct, but they can form homo and heterodimers as well. They help set resting membrane, they regulate solute balance, and they're really implicated in a number of diseases, both in pain perception, epilepsy, anxiety. These are three areas that we have been interested over the last several years with our compounds. Also, reward and addiction. And the class we're looking at is actually the KIR3 or the KCNJ3 sort of gene, 9, 5, and 6. So you have KIR 3.1 to 3.4. We also have a pretty closely related, this KR 6.1. These are sort of further away from the others. So when we're looking at selectivity of compounds, these are one area that we need to be testing against. So these are encoded by the larger KR family, as I said, the 3.1 to 3.4. They do have homo and heterotetramers. So GIRK1 does a heterodimer with the 1,2, 1,3, 1,4, and then 2 can do those as well. They're widely distributed in the central nervous system, especially the GIRK1,2. And then certain subunits have more narrow distribution. So the GIRK1,4 is in the heart, and the GIRK2,3 is in the VTA. They link to a variety of diseases, as I said, and you can kind of see this complex here. It's now over 10 or 11 years old. There is a cryo-EM structure recently, so that's been somewhat helpful in looking at these compounds, designing compounds. And you can see sort of the different combinations, and the GIRK1 does not exist on its own. It only exists in these dimers, specifically mostly 1,2 and 1,4. And again, you can see the cortex, hippocampus, the cardiac atrium, and then the VTA. So each one has its own individual sort of disease state, where the GIRK2,3 would be thought of for this reward and addiction. So we started this a number of years ago when I was still at Vanderbilt, and we wanted to identify these GIRK activators, and you can see sort of the trace of these compounds. So non-selective, you can see the activation of the GIRK1,2, but also activation of other channels, and then a selective one would be here with no activation here in a GIRK. We found about 100 selective, about 200 non-selective, and the best activator hit we had was this initial compound, this urea. This urea with this pyrazole was really a privileged scaffold for a number of years. We could not do anything to the urea, and that causes a lot of problems because ureas have poor solubility, they have poor brain penetration, and if we're looking at a CNS indication, that's really can be challenging. But as a starting point of 1 micromolar, we were able to do some medicinal chemistry around and are able to identify this ML297. It's about 160 nanomolar, so we have about a 5-fold increase. Solubility is not terrible in this, but the half-life is very short, 20 minutes, and you can see the brain to plasma ratio is 0.2. So it's really fairly peripherally restricted. And it gets metabolized at this methyl group by P450, and that's inactive. So there's still, even though this was a proof of concept compound, there still remained a lot of improvement that was needed. But like I said, it was a tool compound, so we were able to dose high enough to be able to get brain concentrations to be able to see effects. So first is motor activity, motor coordination, and then condition-place preference, which is a study for reward and addiction. And up to 60 mg per kg does not have a significant effect on motor coordination, and does not show a dependency in this model. So we're able to use this, look at it in a couple different pain models. First, in this spared nerve injury for neuropathic pain, it showed comparable activity to gabapentin. And then in this CFA-induced acute inflammatory pain, you can see this response here. We didn't have a positive control here, but this is a pretty straightforward acute pain model. So ML297 set the stage for us, gave us the preliminary data needed to like all of it, as academic, to to get funding. So we're able to get an R01 to design new molecules with better properties. So as I said, this urea had been privileged for a number of years, and we just really could not figure out how to overcome this. And I'm going to show you a lot of different mimics that we had, potential bioisosteres. Not a single one worked. And these are not just singletons. Out of each of these, there's probably 50, 30 to 50 compounds made. So we really did a lot around here, and just, going from these typical urea type of isosteres to now a simple amide, whether it be reversed or not, going to the reversed amide with a single carbon spacer. And we're able to finally find one. If that's just simply kind of taking this nitrogen out. We changed from the phenyl to a benzyl, but that was a known change. And 2 micromolar, not going to really get you too excited, but again, as a starting point, we thought we could do something with it. And my experience now, about 20 years, it's pretty rare that a project cannot be advanced simply because of potency. It's usually a lot of other things. So we were pretty confident we could get this into a potency area that we needed. And you can sort of follow along here over the past about 8 years. We took this 2 micromolar, just made a simple change from this fluorine to a chlorine, really gave us about a 4-fold increase in potency. Now we're at about 500 nanomolar. And then taking this benzyl to the cyclohexyl gave us another couple fold increase. But more importantly, it really helped us in brain penetration. So now we have a brain to plasma ratio of about 0.9, which is pretty acceptable, especially for a tool compound. We published this first in this ACS Chem Neuro, that's the chemistry part, but this British Journal of Pharmacology in 2019 took this compound into a number of different animal models, not for pain, but for anxiety. So stress-induced hypothermia was one, and it showed really nice effects. But we still had an issue with intrinsic clearance. This amide bond gets cleaved by amidases, and it just doesn't have a hugely long half-life. It's long enough to do some acute studies in vivo, but not something you would take forward. So in the process of this, we found this tetrazole compound, and with 8 micromolar, but we know we didn't have that right, or the left-hand portion that we needed. We were able to now get under 100 nanomolar with this middle compound, but just again, really poor plasma stability. We did notice when we put this cyclic sulfone on, we lost all activity, but we improved it. So we thought, well, let's look at this again, and we're able to now get to this compound. We published this a couple years ago, about 140 nanomole, but now we have actually an improved metabolic stability compound. And you can see some of the properties of this compound. We have some selectivity against 1,4. Said earlier, it's in the cardiac, so as a drug, we would definitely have to improve on that, but as a tool compound, we think we're okay. Selectivity against a number of other ion channels, selectivity against a 45 panel receptor. And we're pretty pleased to see that in a in vivo PK study after IP dosing, we have really good brain plasma ratio. So brain to plasma ratio is about 2.0 with about 2-hour half-life. So much improved PK over the original compound. So we've been able to now take this into a number of different compound or models. We do the mechanical pain in the spared nerve injury model in mice, and we see a analgesic effect, dose-dependent analgesic effect, and one that's on par and actually significantly better than gabapentin, which is a sort of a clinical compound used for this. So you can see at 30 mg per kg, we have a pretty good response. Again, you're starting to lose that response after 4 hours just because of the half-life. We also look at a number of different things. We look at this MIA model, so the monoiodoacetate, which is a model of osteoarthritis. It's a well established, and you can see the response we have again. And compound 3 is still A18. We just changed it when we were doing some publications. You can see the response. You can see the weight bearing as well as a neuropathic component. The model does due to the peripheral neuronal. So it produced analgesic effects and improved weight bearing distribution as well. And you can see this 1 hour post dose. And then we also looked at this in chronic compression of the dorsal root ganglion, the CCD. So this is sort of a sciatica and low back pain model. And we can see now to get really good response, we need to go up to 50 mg per kg in gabapentin. And our 30 mg per kg still gives a really improved response for that, up to 2 hours. So we have, 30 mg per kg exhibited pain threshold similar to the pre-surgery baseline. So now we've shown multiple models that our tool compound works in. So we now, I think, have really good evidence showing GIRK1/2 as a potential therapeutic, or at least a preclinical for pain. These compounds are not novel, they're not proprietary. So if we're going to take these into more drug discovery, we're going to have to design new compounds. The other thing is I did not show in this, we also did it in the condition place preference, so that model of addiction, and we do not have any effects in that too. Now, that's not the gold standard. The gold standard would be self-administration. But one of the properties of this compound is solubility still is not where we want it to be. So it's challenging to have our compound as self-administration, where you have to have a liquid for the animal to come in and press the lever and get the dose. So we use the condition place preference because we can dose that IP. So we've shown, I think, that GIRK1/2 activation to be implicated in pain processes. Compounds from our lab are active in multiple animal models, multiple pain models, but also we've shown some to be in anxiety. We had extensive medicinal chemistry optimization, and that identified this ether-based, which improved PK and selectivity profiles, and then shown these in models. So further work is planned. We're still looking at a full mechanism of GIRK1/2 activation in different pain models. We're getting ready to, as was said, some of the work on newer compounds we just published in ChemMedChem, and we're looking to publish the full pain profile soon as well. The real challenge with looking at these compounds to get novelty is not just the amide portion, but that pyrazole, and I really didn't highlight that. This pyrazole is absolutely required. We've been able to try other 5-membered rings from just a parole. We've substituted to a thiazole. We've substituted to imidazoles. We've actually made this a 6-membered ring with the nitrogen here. We've actually tied this back into like an indazole. Everything we do out here leads to complete loss of activity. So this is really a part of the molecule that is not amenable to change. So we're looking at other things. We're looking at macro cyclization and other techniques to try to get to maybe some novel compounds. So a project that's been well established in the lab and has been well funded by the NIH as well over the last several years. The next one, we're going to continue with pain, but this is really born out of selectivity of one project leading to another project. And this is from dopamine 4 to sigma 1. We've been looking at dopamine 4 receptors for a number of years. We have an active R01 for this through the NINDS, looking at this for Parkinson's disease. So again, a number of chemists in my lab, graduate students and postdocs. The animal model is run with Dr. Angela Chinchi at Lund University in Sweden. And then we have utilized the psychoactive drug screening program at University of North Carolina, which is free to NIH investigators. They do 45 CNS important receptors free of charge. So as I said, dopamine's been of interest in our lab, and the D4 receptor is not nearly as well understood or studied as the D2, D3. D2, D3 is sort of highly implicated in schizophrenia. It's sort of the dopamine hypothesis of depression. So D4 is less widely available, I guess, than the D2 and D3, but it is somewhat restricted too. So in those areas of the brain, even though it's not as expressed as much, it still could be beneficial. So the compound in the middle, this L745870, was actually taken into the clinic. It made it to phase 2 clinical trials for schizophrenia, and it failed for efficacy. However, it was well tolerated. So it gives us some confidence that if we can find a D4 compound, we think it should be safe. But when they started looking at it and looked at the profile of this, you can see it's a very highly active against D4 as far as a Ki value. So they looked at this because D4 is expressed in the striatum, which is where a lot of the dopamine neurons are lost in Parkinson's disease. So as you lose those neurons, you start to have to replace dopamine. That's the major clinical drug is L-DOPA. And as you take L-DOPA, you get more and more issues with it. So they showed this 745 did show benefit in a this non-human primate model of PD, although it failed to improve rotarod performance. So they thought that the therapeutic index would be too small to move forward. But again, it's a proof of concept in this. So when I was at Vanderbilt, we found a new synthetic procedure for making these chiral morpholines. And, to be honest, we weren't really sure what to do with these chiral morpholines. So we sort of looked at the patent literature and saw that compounds like this had been tested in D4 models back in the mid-90s by Merck. So we just looked at these and sent a lot of these out for activity. And sure enough, we were able to get a pretty active compound, 14 nanomolar Ki value against D4 and fairly selective against the others. In vitro PK modeling showed pretty good free fraction. You see high clearance in a rat, unfortunately, and we know it gets this phenyl ring gets oxidized as well as this nitrogen gets dealkylated. But good brain penetration. We can see we have about twofold brain concentration above our Ki. We took this through the program in the NIH to look at for addiction models, and they showed that it's clean against the Ames and does not have effect on locomotor activity. So we're pretty happy with this compound. But again, we had some issues to solve taking this into a drug discovery program. But part of this, we wanted to see, does our compound compare to that L compound or the Merck compound in a model of L-DOPA-induced dyskinesias in mice? And Angela's group at Lund compared our two compounds. So the model is, it's a mouse model, and then you give them L-DOPA over a number of days, and they start to show these dyskinesias. These dyskinesias are involuntary movements, head twitching. We also look at sort of the oral facial muscles because Parkinsonian patients sort of lose some of that muscle tone. They have about 8 to 10 different markers that they look at, and they add all these up, and you can see the dyskinesia score up here, about 25. And you can dose our compound in a dose dependent, either 10 or 20 mg per kg, and you can see the reversal of the dyskinesias. And you lose all dyskinesias after about 2 hours. And that's, again, L-dopa's half-life is maybe 90 minutes or so. So it's hard to keep them dyskinetic for too long. So, ours loses that too, but that's mainly because the mice are no longer dyskinetic. But you can see we have a pretty dramatic reduction. Our 10 mg per kg and 20 mg per kg was 60 and 70%, compared to that Merck compound, which only showed about 12 and 22%. And you can see the dosing's different. It's 1 mg per kg and 3 mg per kg. And what we tried to do is match the PK exposure to the dopamine activity. So we would keep that same. We weren't overdosing anything. Keep that same 2 to 3-fold above the Ki value, and tried to match that head to head. We also showed some other amplitude scores. So we have a pretty good dopamine compound, but we had some issues with the morpholine. So we decided to do some chemistry and just take that oxygen out. Now we're going to the piperidine, or we sort of move this to the 4 position. And you can see we had some reasonable success, but we did lose a lot of activity. So from the morpholine to the piperidine, or the oxopiperidine, the 4 or the 3. You can see we went from 5 nanomolar now to somewhere in the 100. So we're losing 20, 25-fold selectivity. But when we did our selectivity panel, we kept seeing this sigma-1 receptor, and kept seeing pretty potent compounds in the single-digit nanomolar binding. So this piperidine, the 3 piperidine was 2 nanomolar, the 4 was 5 nanomolar. So we decided to dig in a little bit more and see what potentially a sigma receptor modulator could do. And there's really 2 different things that sigma receptors. So sigma-1 agonists are being looked at in cognition models for Alzheimer's. And sigma-1 antagonists, or sigma-1 inhibitors, are really looked at for pain. And you can see a lot of different areas of the brain, both in the, say, microglial, the spinal cord, the dorsal root ganglia. All of these sort of are where sigma-1 receptors could be present and be active in these models. And several sigma-1 receptor antagonists are being evaluated in preclinical models of pain. There's Dr. McCurdy's group at Florida has published quite a bit. And there's actually a Spanish group that took a sigma-1 receptor antagonist into clinical trials. They reported the phase 2 results about 6 years ago. I've not seen anything since then, but it was well tolerated and showed activity against neuropathic pain. That's the most furthest along sigma-1 receptor that we know of. So obviously, even if you have a single sigma-1 receptor, there would always be room for improvement or additional compounds. So just looking at these compounds, as we move along, we still maintain this sigma-1 activity. And I have the D4 just as a comparator, that we are getting separation between those two. So what started as a D4 project now has kind of morphed into a more selective sigma-1. But you can see some interesting SAR trends, and that is this benzimidazole has a significant fold difference between sigma and D4. If you put a methyl on here, you gain that dopamine receptor activity back. Same thing with the indazole. You add the methyl, you gain the D4 without significantly changing the sigma-1 at all. If you go to heteroaromatic substitutions in the northern part, you're losing sigma. And there's a crystal structure of sigma, and we've docked our compounds, which I'll show in a little bit. It doesn't really explain these observations, so we're still working on that. As a chemist, I need to show how we make these. They're pretty straightforward. You just alkylate the hydroxypiperidine, remove the Bach, and then either alkylate or do a reductive amination. So it allows us to make a lot of compounds, which is nice. And then just going through some of this SAR, I won't sort of belabor it, but you can see the southern portion on the sigma really does tolerate a wide variety, but slightly different as well. So this indole is very potent. If we go to this imidazopyridine, we now lose to 143 nanomolar. We can gain that back if we just do the regioisomer in a methyl. And then this regioisomer as well, we gain it back. All while maintaining, you know, a 15 to 30-fold or more selectivity against D4. You can see some of these other changes here as well. Having this 3-piperidine, we thought maybe we could tie this back into a cyclic compound that might give us some novelty, novel compounds to move forward with. And we wanted to really keep this oxygen or a nitrogen we thought could mimic this as well in that position. You can see this 6-5 system, or if we reverse it to the 5-6 or the 5-5, they all maintain sigma activity. Interestingly enough, they all lose dopamine activity. These spirocyclic compounds are not active at dopamine at all. And they really have a really good selectivity profile against a wide range of compound or of receptors. And you can see here as well in this 5-5 systems really striking. So we're able to, you know, find a number of compounds with good activity against sigma-1. Sigma-2 is a closely related. In fact, the sigma-1, sigma-2 receptors were only really fully isolated about 30 years ago. Originally, when they were identified in the 70s, they were identified as an opioid receptor. And then with further work, they're able to separate those out from the opioid receptors. But sigma-2 is also, inhibitors are implicated in pain and being advanced for pain. So we don't think the sigma-1, sigma-2 separation is necessary. In fact, it might be beneficial, but it doesn't have any activity against the other dopamine receptors. And the compounds 1 and 2, our original tool compounds, have no significant activity against this receptor panel. You can see some of the other properties here. We have pretty good human intrinsic clearance. This one, less than 20, less than 10, is considered stable. So when we look at this hepatic clearance for the intrinsic clearance, you're looking at the hepatic blood flow. So for humans, it's roughly 20 mils per min per kg. So we're about half of that. And then for mice hepatic blood flow is about 90. So again, we're about half. It's hard to, my experience, it's really challenging to get a really stable compound in mice. They have a lot of SIPs to chew up compounds. But again, these are tool compounds, so all we really need to do, as you can see from all the pain models, are out to about 4 hours. So we don't need, you know, sort of once a day, daily dosing. Look at free fraction, human and mouse, very good free fraction. So if you're looking at that free drug hypothesis. And then when we dose this into an in vivo study, you can get half-lives of 2 to 4 hours in plasma, but a much longer half-life in brain, which is a little concerning that it's not the same. It's easy to look at that in a mouse. It'd be a lot more challenging in a human to do those types of studies. So really would like to see that matched a little bit better. But again, for our purposes, we can bring these forward in these animal models, and then the brain to plasma ratio is 3 and 5. And this, what's known as KPU, is an unbound plasma, unbound brain. So again, speaking to that free brain hypothesis. So we have very high levels in the brain. These are passively permeable. We did, like I said, we looked at some of these docking, and nothing really stood out as to why we see the selectivity that we do. The pharmacophore is very similar for both. In the dopamine, you see this key hydrogen bond with the nitrogen, your basic nitrogen, with the asp 115, and then sigma, that's a glue 172. That's really the anchor for all of these. And then the rest is just kind of filling pockets with these greasy aromatics. If you dock these in, you can get this pose. It flips and puts this up here and this down here, just because they're almost symmetrical in some ways. And then you can see this spirocycle, which lost all activity against the dopamine. We have it docked here in the sigma, but we dock it in the dopamine. It still scores well. So we really, I think, need someone more talented than I am to do a little bit more work in the modeling. I think we need some molecular dynamics and other calculations as opposed to just simply a dock. So our lead compound is this SSA27, which is a slightly different structure than the ones I showed before, but in the same general structural class. You can see, again, we did this in the spared nerve injury model of mice, and gabapentin is in the light blue, and our compound at 30 mg per kg in red is statistically significant, seeing activity up to 2 hours, and even up to potentially a little bit longer than that. Pain medicine is another one that's you don't really have to necessarily go for once a day dosing. There a lot of these pain, if you take Advil or Motrin, you're taking it multiple times. Ultimately, we'd like to get a once a day dose, but that could be very challenging. Again, some of that's just the pain that each individual person feels. And this is with the collaborator Simon Shih out at AFAC up in Palo Alto. So we had this, and part of this is, we have graduate students that need to be an organic chemist, not just a medicinal chemist. So we wanted to really find new structural scaffolds to look at and see how we can make those. So from these spirocycles, Enamine over the last several years has really published some interesting chemistry to make these scaffolds. So scaffold A, whether it's this bicycle, this here is a 330 bicycle, or this spirocycle, published a couple different papers, and then this really interesting sort of 311 structure that was published in 2023. We thought, again, we modeled these. We thought that we could make these, and actually by putting the rest of the sort of stuff around these compounds, we would get some novel compounds, and we've been able to submit patent applications on these compounds and have these submitted for that prior to us publishing. The way these are made pretty straightforward. You have these cyclic alpha beta esters. This sort of reagent breaks down into this 3+2 reagent, and you just do a 3+2 cycle addition to get here, and it just depends on what the ring size is of this. We've done the 5-membered ring, we've done a 6-membered ring to give us sort of a 330, 340. We've also got some 3-membered rings here, so just a 310, but we make that in a different manner. So once you're here, you can just do a reduction of the ester, mesylation, and then you react with the phenol, take off the benzyl group and put that on. Similar chemistry to do the spirocycles. You do a Wadsworth-Emmons reaction, 3+2 to get here. What's interesting, we can take this forward just like we did above and get to these compounds, but if you reduce this ester in the presence of lithium borohydride, once you get the alcohol, it then attacks this, opening them up, this oxetane, to now get this 330, although with the oxygen, which is a really interesting way to diversify our chemistry without from the same starting material. We're looking at sort of some mechanistic understanding of why you really do need the lithium borohydride. Probably the boron is chelating here, and then that helps to open it up. But if you take that away, you just get simple reduction. And then this 3-1-1 system, you can make this from this tribromo alcohol, deprotonate the alcohol, and it reacts here to get to the oxetane. Variety of whether it's a methyl, phenyl, ethyl, it doesn't really matter what this is. You reduce this, you get to this intermediate, and then this nitrogen attacks here, opens this up to get to these sorts of ring system. So it's a good way, as chemists, as my students, to think about new chemistry. But are they active? And the answer is yes. The 3-3-0s, again, predominantly preferring the sigma 1. These 3-1-1s are a little bit more, I'd say, equipotent, threefold maybe for sigma, but it depends on what you put down here. And the spirocycles now, these are the ones that actually now come back and get us the dopamine, albeit in modest selectivity. But now we're now into the pretty potent KIs of 13 picomolar and 60 picomolar. So we're pretty excited about these. We have done some preliminary stability, and they're not very stable, but we're hoping to be able to fix that as well. So for this, we discovered a piperidine-based scaffold, potent selective sigma 1. I call them modulators again because we really are looking at binding right now. We have to look at a functional assay. It started from our D4 antagonist program and just looking at selectivity and see where we could go. PK studies show they have modest half-life in mice, and we're trying to again determine the functional activity of these compounds. We call them in vivo functional antagonists, but we need to fully do that in some more in vitro studies. And with that, I just always want to thank my lab. I've got a great group of students here at Nebraska, and as always, funding. It's challenging right now and somewhat nervous right now, but we've been able to be lucky enough over the past few years to have really nice NIH support. And then with that, I will be happy to take any questions anyone has. And thanks for the opportunity. I really appreciate it.
You can ask a question through chat, or you can ask straight away. Dr. Hopkins, I have one question.
Yeah.
Were you using any of this latest AI type of applications for predicting physical properties of these drug candidates?
Yeah, and I think that's going to be the next big thing. I think there are a lot of really good programs. I think, unfortunately, there aren't a lot right now that are publicly available. So a lot of my colleagues that are in industry, they present and show these beautiful models of whether they be PK predictions or metabolite predictions. I think that's going to be the next big one, is if we can get whether the NIH or someone like that would support a public database that we could use. I think AI is going to, there's several ways AI is going to revolutionize. I think the very first one is going to be in clinical trial design and choosing the right patients. Most times when compounds fail in the clinic, it's because of the clinical trial design. And then, you see a lot of companies that are getting a lot of startup money because they say we're gonna, find a drug candidate in 6 months. But where it's more interesting too is the FDA just came out and said they are not going to require animal toxicity studies, and they're going to rely on AI to do that. So I've not seen the successes yet, but I'm sure companies are working on proprietary uh, uh, algorithms to bring that forward.
Mhm. Can any of the alkaloids be used as model compounds?
Any of the alkaloids? It's a good question. I'm not sure.
Well, the opioids are the alkaloid type, but some other type of systems, not the opioids, but some other systems.
Yeah, I'm sure yeah. Right now we use the opioids because they're clinically relevant.
Mm.
But yeah, and there's a lot of work trying to also still look at the opioids, but sort of have these biased signaling, right? You have the G-coupled signaling pathway versus the beta-arrestin pathway. And if you can decouple those, the beta-arrestin's the bad and the G-coupled's the good. There is compounds in the clinic right now looking at that. And so you can just take what is good about the pain relieving and get away, not only tolerance, but right, it's the respiratory depression that causes the issue, right? That's what you're going to have. If you overdose because of the respiratory depression. Uh, and then the other is, opioids cause significant constipation. So GI motility, we have to do a GI motility study for ours for the NIH just to prove that, we don't have that issue as well.
Okay, very good. Well, if there are no other questions, it is my present duty to actually thank Dr. Corey Hopkins for a very good seminar, and it's for organic chemists, it is a treat. I am also an organic chemist, so it is definitely a treat. And I hope that in the coming near future, you will find a lot more success in developing compounds that can be actually clinically be used and prescribed by physicians. Thank you, Dr. Hopkins. Thank you very much for your time and for your very nice and interesting seminar.
Thank you. I really appreciate the invite. Have a good rest of your evening, I guess.
Thank you. Thank you.
Thank you. Have a good night.
Okay,
Thanks.
thank you all. Thank you.